Haploid cells containing just one set of chromosomes, so they've got one of each chromosome, and we'll see what chromosomes are in a second. But these are basically packages of DNA wrapped around protein. And the DNA is where we'll find the genetic information. Not all of the DNA consists of what we call genes, pieces of DNA that code for characteristics, or more specifically code for certain proteins. But haploid cells contain only one set. So our other body cells, each of our body cells, our skin cells, our eye cells, our hair, whatever cells we're talking, liver cells and kidney cells, they all contain two sets of chromosomes, one full set from one parent and another full set from another parent. So what we talk about, the haploid number is 23. That's the one set of chromosomes worth, if you like, of human DNA. And because our body cells get twice that, then we refer to diploid cells, or diploid cells rather have 46 chromosomes. So those are what we call the somatic cells, the body cells, okay? So we're going to be looking in detail at this series of events called the cell cycle. And the cell cycle consists of 3 stages, okay? Mitosis, or nuclear division, whether it's mitosis or meiosis, is only one part of that cell cycle. In fact, it's a pretty short part of it. Most of the time, the cell is just going about its regular business, right? It's growing, it's getting bigger, it's metabolizing, it's producing all the enzymes it's supposed to do, it's making all the necessary molecules that it needs to make to do whatever it is it's supposed to do, whatever its job is. If it's an eyeball cell, then it's going to be producing, whatever eye cells need to be producing. If it's a liver cell, for example, then it's going to have a lot of mitochondria because it's really busy, and it's doing a lot of chemical reactions, metabolic reactions. But all of that refers to what's called interphase. An interphase is just the regular life of a cell until it comes to a stage where it's got big enough. It's a certain stage or a certain age, let's say, and some sort of trigger has caused it to enter the stage where it needs to split in two. Right? Before it can split in two, though, what it's going to have to do is, because it's got because there's a nucleus involved in these eukaryotic cells that we're talking about the cell cycle of, the nuclei need to split in two first of all. So if you have a situation where you're going to split the nucleus and it's going to be shared between 2 different cells now, 2 new cells, well, surely that would mean that the amount of DNA is going to have to get split. And surely that can't be a good thing. That would mean one cell would only get half of the required amount of DNA, and the other cell would be the same. So we've got a problem there, first of all. We can't just split cells in 2, because each time we did that, we'd end up with half the number of chromosomes in each daughter cell, right? So we've got a bit of an issue. So we'll see how that works out in a second. Once the nuclei have divided, then the cell can finally split into 2, right? Because each of there are 2 nuclei that can be one can be provided, if you like, for each of the 2 new cells. Okay, so 3 stages in the cell cycle. This is the kind of picture of it. You can see the interphase is how cells spend most of their life in that phase, and they, like I said, it's the normal life of a cell. However, one important thing to answer the question I was just asking, one important part of interphase is that at some stage towards the end of interphase, there's going to be a replication of DNA event. What that means is all the DNA is going to be copied. And now we start to be able to make sense of what's going on. Because now if you've got only, the cell has its full set of DNA and it's going to split in 2, well, if that's going to work, then all of the DNA has to be copied first, so that there are 2 full copies of the DNA just before the cell splits in 2. Just before we can have the nuclear division followed by cell division, we've got to have this DNA replication. All the DNA has to be copied, because one new cell has got to have a full complement of DNA, and so does the other new cell. So we've got to have double the amount of DNA so that each cell is going to have its, the full amount. So that's the DNA replication. That's a kind of an important thing to remember, because otherwise this doesn't really make sense. Otherwise, every time you have or split the cell, then you'd be reducing the amount of DNA. So that's to avoid that, right? So what happens in interphase then? Like I said, they're just doing their thing. They're doing their job. They're actively growing. They're metabolizing. They're making new cell organelles as they get bigger. They're producing the enzymes that are needed to do all that. In order to do producing the enzymes means that they're doing all the protein synthesis stuff at the ribosomes we talked about last week. They're doing transcription of the DNA into mRNA. First of all, we talked about that as well. And then, like I said, they're translating that mRNA at the ribosomes. We're going to cover all that protein synthesis in detail later. And then, like I said, then the DNA is all copied. Okay. Now, one important thing to point out to you is that when the chromosomes, and these are individual, let's call them individual packages of DNA and wrapped around protein to kind of keep them packaged. Okay. But the DNA is the really important part. The protein is kind of incidental. It's just packaging. Okay. So if you can imagine a single strand of DNA is a chromosome. It's not a single strand. They're double-stranded molecules. Okay. But one single chromosome, which is made of double-stranded DNA, all DNA is double-stranded in humans. So this is a very long piece of very tightly coiled double-stranded DNA, but it's a single chromosome. Now, during DNA replication, each of those has to be copied. So a full copy is made, but the copies end up being attached. Okay, so you can imagine these are just copies, literally like replicates of the original strand. They're both identical now, and they are held together by a structure called a centromere. Okay, so I'll show you that in a second. We refer to each of those copies, each of those chromosomes. So one chromosome, then you get the copy, which is attached to it. This is what we still call this a single chromosome, even though it's got this full copy attached to it. We call the structure itself a chromosome. It consists of what we call two sister chromatids. Okay, so chromosome, towards the end of interphase, that chromosome is going to be copied, and it's going to be its copy is going to be attached to it at the centromere, and we're going to call this a chromosome now. It's an X-shaped sort of thing, and it's going to be held together at the centromere. Okay. Let's just have a look at a couple of questions here. If we took a single cell from a multicellular organism and supplied it with glucose, would it be able to stay alive, or does it need other similar cells? Oh yeah, for sure. So that's done in tissue culture all the time. It would need more than glucose. You'd need to provide all it would be complicated. But yeah, sure. Human cells, individual cells can be taken from humans, from all kinds of organisms, and grown in a petri dish, as long as they're supplied with all the nutrients and factors and everything else that they want, and the conditions are perfect. And that will not only stay alive, but you can get that to divide and form tissue. So we do that. That's kind of routine these days. Another question is, how often does a normal cell cycle last? I don't know if there is an answer to that. So some are very fast, rapidly dividing. It depends which organisms you're talking about. It depends which cells you're talking about in a tissue. So that's a Google question. So it would depend. If you want to know particular, how often do liver cells divide, and you Google that if you want to know how often other cells, and it does very much vary. So some are very, it's quite a quick turnover of cells, like skin, for example, or liver, or some are not at all. Okay, so let's just have a look at this in sort of picture form, or in an animation form, rather than me just trying to describe it with gesticulating with my hands and things. So have a look at this. I think this is quite good. I'm going to skip to this section here, and just notice that it reminds us that there are three phases of the cell, three parts, three stages in the cell cycle. We've got interphase, mitosis, or meiosis, right? But mitosis in this case, and cytokinesis, which is the cell splitting in two in the end. So if we look at the beginning of this, we just focus on interphase. I think it's worth just having this kind of image in our head. So what we're talking about here in interphases, the normal cell life, as we said, and cells are just doing their thing, they're growing and whatever, carrying out their metabolic activities, their job, right? And inside the nucleus, the chromosomes are not in these. If you were to look at a cell under the microscope when it's in interphase, you can't actually see the chromosomes as these tightly packed, obvious individual structures, right? They're too loose. So the DNA and protein is much more stringy. It's kind of spaghetti-like, and it's all kind of tangled. One chromosome is tangled up with the other chromosome. You can't see them as individual packages. In order for that to happen, as the cell, and for us to be able to see those individual chromosomes, as the cell enters the first stage of mitosis, we'll see that all the DNA protein, what we call this chromatin, right, is the version of it as it is in interphase, this loose, stringy sort of state that the DNA and protein is in. That will tighten up. It'll do what we call condense into individually visible, under the microscope, packages of DNA and protein that we kind of refer to as chromosomes. Okay, so now the chromosomes are in this chromatin form. So that's this stringy DNA they're talking about here. And as the cell enters the first stage, and we'll just pause it there, and we'll go and have a look at a couple more ideas or terms we need to know. This is where the DNA at the very beginning of mitosis will start to what we call condense. Okay. But what this is showing, and I just want you to see this, is that each of those chromosomes is actually attached to a sister chromatid. Okay, and they are identical. So the one on the left and the one on the right, they are that's a pair of individual chromosomes. This cell here has 4 chromosomes. Okay, but you notice that each of the chromosomes consists of 2 sister chromatids. Okay, okay. Now, just to confuse things even more, what I'm going to say to you is that this cell here has got 2 pairs of what we call homologous chromosomes. Now, what we're talking about, homologous chromosomes, it's nothing to do with those sister chromatids I was talking about. The homologous chromosome idea is that there are 2 pairs because, so let's say that one set of chromosomes from one parent are blue. Right? There's 2 blue ones in there. So in this cell, this is a 4 chromosome cell. It's diploid. So it consists of 1 pair of chromosomes from one parent and another pair of chromosomes from another parent. Let's say the blue ones are from one parent and the red ones are from another parent. So there are 4 in total. But what we talk about here, there are 2 pairs of homologous chromosomes. Homologous meaning 1 from one parent, 1 from another. Now, they're not identical. They're going to be very similar, those 2 in the pair. But there's going to be differences in them, right? The differences between one parent and another parent. The sister chromatids are identical. Practically, anyway, let's say they're identical. Because they are literally just copied during DNA replication by an enzyme. They just come along and copy exactly the DNA. So there's a difference, right, between sister chromatids, which are 2 copies, or 1 chromosome and a copy of it attached to it, and homologous chromosomes.